In this video, we're going to talk about the spermatogenesis in male reproductive system. In the previous video, we have described the anatomy as well as the histology of the male reproductive system. So basically, in this video, we're talking about spermatogenesis, which is a little and a very cool and interesting process that occurs in the male reproductive system. And basically, if you're talking about spermatogenesis, it means sperm production. You understand? Like spermatos, sperm, and genesis, production. So the production of sperm is referred to as what? Spermatogenesis. That is just the what? That's just the basic definition of spermatogenesis. But it involves a lot of processes, like a lot of cool and normal process. You understand? So I will just be analyzing the process of what? Of spermatogenesis. Now, let us look at the specific organ. Let us look at the specific organ around this place. Let me put this. Now, looking at the specific organ around this place, we have something like this. Can you see? So, this is the hypothalamus, which is the master endocrine regulator because it controls the pituitary gland. You understand? So, let me just zoom in. Let me just zoom in into this hypothalamus. So, look at this. This is an hypothalamus. This is what I drew there the other time. Now, this hypothalamus consists of a bunch of gray matter. You understand? And a specific set of nuclei. And this nuclei is responsible for the secretion of an hormone known as gonadotropin releasing hormone. So it releases what we know as GnRH. You understand? So gonadotropin releasing hormone. That is what it releases. And remember, I told you that it consists of specific set of nuclei. And this nuclei are just two. We have let us call this one preoptic nuclei, PO nuclei, preoptic nuclei. And let us call this one aquatic nuclei. Understand? Aquatic nuclei. So these are the two nuclei that we have in what? In gonadotropin. Now, this gonadotropin releasing hormone now bind with a little cell down here. Look at this little cell. Even though it is not very accurate, I just to draw it somehow. So look at this little cell down here. This little cell is gonadotropes. You understand? It is known as what? Gonadotropes. You understand? That's what we know as gonadotropes. Now, these gonadotropes stimulate the production of two hormones, two basic hormones, and these hormones are the luteinizing hormone, represented with LH, and follicle stimulating hormone, so FSH. So these are the two hormones that these gonadotropes triggers. You understand? It triggers the production of what? Of luteinizing hormone and what? And follicle stimulating hormone. And that is what I actually indicate in this slide that I showed you the other time. So I showed this particular slide. So look at this. Look at this. So in this hypothalamus, this is the hypothalamus, which is the master control gland. We have the preoptic nucleus and the aquatic nucleus, and this is the gonadotropes. These are gonadotropin, that is GnRH, which is stimulated by this nuclei, stimulates the what? Stimulates the gonadotropes. You understand? And these gonadotropes basically function in the what? In the production of two hormones, or secretion of two hormones. You understand? And that is what? That is the follicle stimulating hormone and the luteinizing hormone. I'll tell you the function of these hormones as we are moving on. Now, let us consider a big structure down here. Look at this big structure that I just drew down here. So this is a big structure. This structure is known as the seminiferous tubule. Remember, I told you that inside the testis we have about 1 to 4 highly convoluted seminiferous tubule. So these are seminiferous tubule. I'm just trying to zoom in into one of the seminiferous tubule. You understand? So what the importance of this tubule is that this is where the sperm production occurs. So inside this tubule we have what? We have where the sperm production occurs, basically where the sperm production occurs. Now this cells is made up of what? Is made up of a bunch of important cells, and these cells are known as is known as Sertoli cells, Nod cells, or sustenicular cells. Some textbooks call it sustenicular cells. So, but me, I will call it Sertoli cells. Look at this. These are Sertoli cells. You understand? So, Sertoli cells, Sertoli cells, Sertoli cells. So, this whole compartment is seminiferous tubule. But each cell that's inside is written as what? As Sertoli cell, or Nod cell, or sustenicular cells. You understand? Now, it should be important that in between these cells, we have what? We have, look at this tiny hair-like structure that I put here. Look at this tiny hair-like structure. Can you see them? These are these are proteins that link them together to make it to be adherent to one another. So we call it tight junction or adherence junction. So we call it what? We call it tight junction or what? Adherence junction. And we have them around this side. We have them here. Look at it. We have tight junction, tight junction, tight junction, tight junction. We also have one big structure here as well. You understand? It's what? It's adhered together. You know. But one thing about it is just that it is not functioning as only adherence only. I'll tell you the other function of this particular stuff. This stuff. Now look at it. This is the basal lamina. You understand? And we have the basal compartment. We have the basal compartment, we have the adluminal compartment. The reason why we call this one basal compartment is because it's closer to the basal lamina. And the reason why we call this one adluminal compartment is because it is closer to the lumen. So look at this empty space here. This is the lumen. Can you see? I've indicated it here. That is the lumen, and that is where spermatogenesis and spermatogenesis occurs. You understand? Now, let me now show you a structure. So in this stuff, I've shown the basic things that we'll be talking about here. You understand? Now, let me now show you this structure that I drew here. So this is just the basic. I'm just trying to take out one of the satellites, like the seminiferous tubule itself. You understand? So I want to deal with only the satellite cells where the sperm production is actually simulated. You understand? And the lumen. You understand? So remember, I told you that there is a particular junction here where they lay on each other, right? And we have stuff that are here, right? And I told you that in this stuff we have the basal compartment that is marked in blue. I'm going to clean that one later. And we have the adluminal compartment that is marked in what? In orange. Around this place, sorry. This place. This one is lumen. So this is adluminal compartment, this is basal compartment. You understand? Now, like I said we have what? We have this structure that acts as a gate or adherence between the what? Between these two satellite cells. You understand? Now, this junction, what it does is that I'll tell you what it does later. You understand? I told you that it is not only for adherence. I'll tell you the usefulness of this tight junction later. Now, we now have. Let us take for an example. We have a diploid cells with 46 chromosomes around this place. These are diploid cells. N with 46 chromosomes around this place. So let me just try to zoom it in very well. So let me take. I'm having something here. I'm having all these satellite cell stuff. I'm having all these satellite cells. I'm having all these satellite cells. So look at it. So I'm having a what? I'm having N here. Let me say this is the what? This is the stuff that I'm having here. Look at it. This stuff that I'm having here. So let me say these are the stuff. Let me go back to what I'm doing now. So look at it. I'm having this. Do you understand? Now let me clean this. This is I need to zoom it in very well so that I can see. So this is N, you understand? This is N, 2N rather, 2N. A diploid cells. And this diploid cell is called spermatogonium. That's a heck of a name. Spermatogonium, you understand? Now, this spermatogonium undergo mitosis. You know what mitosis is? Mitosis is the cell division in which the parent cells, the daughter cells that are produced look exactly like the parent cells. It means they have the same number of chromosomes. So it is going to separate into two diploid cells. They are still diploid. This one is still diploid. This one is also diploid. You understand? Now, let us call this one primary. Let's call it type A cell. You understand? Let us call it one type A cells. And let us call this one type B cell. Now, what happened is that this type A that is here go back and become another spermatogonium. You understand? So one thing about type A is that it goes back and become another spermatogonium. You understand? So it does not go down, it goes back. You understand? But these type B cells, it releases some type of hormones, you understand, some types of chemicals. And what does this chemical do? These chemicals actually breaks off or tells this particular adherence junction that's open. I want to like undergo division, spermatogenesis. You understand? So this is where spermatogenesis begins. So what happened? These junction, they open. So they open for the what? They open for the passage of the type B cell. So this is the type B cell around here. That is what? That is 2N. You understand? Now, these type B cells now migrate into the abdominal compartment. It migrates into what? Into the abdominal compartment, still 2N. So when it migrates into the abdominal compartment, we cannot call it type B cell again, you understand? We call it primary spermatocyte, you understand? So you understand? I said we call it what? We call it primary spermatocyte. Now, what's the function of this junction? Before I move forward, let me just talk about the function of this junction. What happens is that when does spermatogenesis occur? It occurs at the stage of puberty. You understand? So what happens is that during it might happen before the stage, before the puberty stage. You understand? It can happen before the puberty stage. Now, if it happens before the puberty stage, if it happens before, so as I was saying, I said it may occur before puberty stage. Now, if it occurs before puberty stage, what happens is that, let us say for an example, the sperm cell was able to like enter into the what? Into the lumen, you understand? This sperm cell is going to like release some chemicals, and these chemicals will be taken by the immune system. And once this chemical is taken by the immune system will be like, it is not yet for the production of sperm to occur. And it will be like there is something wrong with this particular hormone. Why is it releasing testosterone? Why is it releasing this? Why is it? So it will cause fights between the what? Between the immune system and the testes. It will cause the immune system to fight our testes. You understand? So we can call this particular junction that is here, we can call it blood testis barrier. You understand? So it creates a barrier between the what? Between the blood and testis, so that the immune system won't be able to fight the what? The testis. You understand? Even the spermatocyte won't be able to like it won't be able to enter into the lumen and stuff. You understand? So like I said once it enters into the abdominal compartment, we call it what? We call it primary spermatocyte. So let me just represent that one. Yeah. So let me say this is what? This is 2N. You understand? This is 2N. We call it what? Primary spermatocyte. Now, this primary spermatocyte now undergo meiosis 1. That is the first stage of meiosis. And once it undergo meiosis 1, let me show that one here. Let me say this is the 2N, right? You understand? It undergoes primary spermatocytes, meiosis 1, to produce haploid cells. It produces what? Haploid cells. An haploid cell is N and N. Now, these haploid cells, they are cells with 23 chromosomes. You know, 2N is 46 chromosome, right? 46 chromosome. And N is what? N will be 23 chromosomes. So this is meiotic 1. You understand? Meiotic 1 or meiosis 1. You understand? Now, this meiosis 1 now proceed. So we call this one secondary spermatocyte. You know, this one is primary spermatocyte. We don't call this one what? Secondary spermatocyte. Now, these secondary spermatocytes, they now undergo meiosis 2. They go into meiosis 2. Now, everything occurs inside the lumen. Now, once they go into meiosis 2 to give what? To give N and N again. You understand? Still haploid. So we can say meiosis 2 is mitotic in nature. You understand? So this is called spermatid. We call it what? We call it spermatid. Are you getting me now? We call it spermatid. Now, these are spermatids. So what happens is that once these haploid cells, the 4 haploid cells has been formed, you understand? They now develop, they undergo what we know as spermatogenesis. They undergo what we know as spermatogenesis and develop into a matured spermatozoa. You understand? They develop into what? A matured spermatozoa through the process known as what? Known as spermatogenesis. So let me just summarize that one here. So I said you're having something like this. Inside the abdominal compartment, we have the spermatogonium, right? We have the spermatogonium, which is 2N, right? It divides into type A and type B. Type A goes back, so this is type A, this is type A, and this is type B. You understand? Still 2N. So type A go back to become another spermatogonium, but type B go into the what? Into the abdominal compartment. Then it divides into what? It divides into 2 haploid. You understand? And we call this one primary spermatocyte. We call this one secondary spermatocyte. This secondary spermatocyte now undergo meiotic 2 to divide into 4 haploid cells. You understand? And we call them what? We call them spermatids. You understand? Now, these spermatids, each of these spermatids now develop into a motile sperm. Look at them. They develop into what? Into a motile sperm. They develop into a motile sperm, and this process is known as spermatogenesis. Now, from here to here, down here, is known as spermatogenesis. You understand? And from this place that I mark red, that is from this place into this, is known as what? As spermatogenesis. I will still be showing that slide. Now, all this stuff doesn't occur on their own. They don't happen on their own. What's the function of luteinizing hormone? What's the function of follicle stimulating hormone? Now, let us go back to the first slide. Let's go back to the first slide that we have here. Now, look at this. Look at follicle stimulating hormone. Look at luteinizing hormone. Now, this luteinizing hormone, can you see the arrow? The arrow, one thing about luteinizing hormone is that it triggers the what? It triggers the Leydig cells. The Leydig cells, also known as interstitial cells. Now, once it triggers the Leydig cells, it goes into the nucleus of the Leydig cells, and the nucleus stimulates the production of an enzyme. This enzyme, it stimulates the what? The conversion. You understand? Or secretes an enzyme which converts cholesterol into what? Into testosterone. So, the function of Leydig cell is that luteinizing hormone comes into the Leydig cell, triggers it. The nucleus of the Leydig cells secretes an enzyme which converts cholesterol into what? Into testosterone. You understand? So, the testosterone now enters into the what? Into the lumen. You understand? It goes into the lumen. So, what's the function of the follicle stimulating hormone? One thing about it is that we have the follicle stimulating hormone entering into the Sertoli cells. You understand? It triggers this particular receptor that is here, goes into the nucleus. You understand? Then secretes what we know as a protein known as androgen bonding protein, also known as ABP. Androgen bonding protein. So, luteinizing hormone stimulates Leydig cells. Follicle stimulating hormone stimulates the what? Stimulates the Sertoli cells. One produces testosterone, the other one produces what? ABP, androgen bonding protein. Now, what's the function of androgen bonding protein? The function is just that it knows that testosterone itself is lipophilic. That is, it likes lipid. You understand? It likes lipid. And something that likes lipid won't be able to what? Won't be able to survive in an area that is consist of other fluid. You understand? So what ABP now does is that it makes the testosterone to be less lipophilic. You understand? It makes it to be less lipophilic so that it can be concentrated around the what? Around the vicinity where the sperm will develop. You understand? And this is stimulated by this particular hormone, luteinizing hormone, and follicle stimulating hormone. You understand? Also, another function of the Sertoli cell itself, these Sertoli cells, another function is that it provides nutrients for the sperm cell. You understand? It provides nutrients for the sperm cell so that the sperm cell can what? Can develop. Now, the testosterone can also go into other tissue, which we won't talk about that now. We don't really need to talk about it. But it can go into other tissue, and the elevation of testosterone is not good for the body. Elevation of what? Testosterone is not good for the body. Now, how does the testosterone, how is it controlled by the body? How is it controlled by the body? Now, one thing about it is that you see these Sertoli cells. These Sertoli cells, they have sensors all around them. You understand? They have sensors that can sense the level of what? Look at this. Let me use blue marker to. So they can what? They can sense the level of sperm in the lumen. You understand? Once they see that the level of sperm in the lumen is enough, what they do is that they secrete a particular stuff known as inhibin. Understand? They call it what? They call it inhibin. Let me write it here. Inhibin. Now, what inhibin do is that it goes into the blood, you understand, then transmitted into the what? Into the hypothalamus. Once it's entered into the hypothalamus, it reduces or inhibits the production of GnRH, that is gonadotropin releasing hormone. Once it inhibits the production of GnRH, what happens to the follicle stimulating hormone? We are going to be having less follicle stimulating hormone, less luteinizing hormone, less ABP, less testosterone, and once there is less testosterone, the concentration of the sperm won't be able to like survive in that particular environment. So there will be less production of sperm. You understand? So it will stimulate the what? The less production of sperm. So look at this. This is just a structure that is actually that is showing the process of what? Process of spermatogenesis. So look at this. So from 46, which is haploid, which is diploid rather, which is diploid, from 46, which is diploid, into another 46, which is the primary spermatocytes. Then we have the secondary spermatocytes when they undergo meiosis 1. Then we have spermatid when they undergo the meiosis 2. Then we have motile or matured spermatozoan. You understand? So these are sperm cells having 23. You understand? So these are paternal. So this is spermatogenesis. You understand? So from here to here, let me indicate it. You see, from everything that is here, like this, from here to here is spermatogenesis. But from here, this place to this place is spermiogenesis, because it is in this spermiogenesis that they become a matured spermatocyte, a mature spermatozoan. You understand? Now, this spermiogenesis consists of 4 stages. You understand? 4 basic stages. Now, these stages are the we have I said they are made of what? Of 4 stages, and these stages are the Golgi phase. You understand? The Golgi phase, the cap phase, the acrosome phase, and the maturation phase. You understand? So we have the what? We have the Golgi, we have the cap, the acrosome, and the maturation phase. So this is GCAM. I call it, this is my acronym for it, GCAM. The Golgi, the cap, acrosoma, and the maturation phase, you understand, of spermiogenesis. So these are the process of spermiogenesis. And in this video, we have be able to like mention the process of spermatogenesis, and we're able to see how sperm is actually produced inside the inside the testis. Thank you for listening, and subscribe for more. We really appreciate you. Thank you so much for listening.